the change in perspective is that we are going now from having several sequences in developability now to having only a single sequence. So that's the big change. In discovery, we have several sequences. Now we need to apply methods to select only one. Then in development, then we have only one selected. We cannot change the sequence anymore. So that's a very big change. And I think historically there has been a lot of work in the literature as well, like modifying, mutating the protein to try to improve the characteristic of the API. But then there is not so much in the literature. Once you fix the sequence, okay, how can we support formulation development now?
What if you could predict formulation failures before ever touching a pipette? Today, we're diving into the computational revolution transforming biologics development with Giuseppe Licari, who is a principal scientist in computational structural biology at Merck KGaA. From predicting aggregation hotspots to designing stable formulations in Silico, Giuseppe reveals how computational approaches are slashing development timelines and catching problems that traditional methods miss. Let's explore how smart science is making formulation development faster, smarter, and more predictable. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Giuseppe. It's great to have you on today.
Hi, David. It's my pleasure to be here with you, and thank you for the invitation to be here in your podcast.
Giuseppe, share something that you believe about bioprocess development that most people disagree with.
Well, in my field of drug product development, I feel that we should set a good enough standard for stability for our API in order to assure that we deliver the products to the patients in a safe way, of course, and in a timely manner. And we don't really need to maximize the shelf life stability, at least for preclinical studies or phase 1 studies. So I think people might disagree with this. They might try to always maximize the shelf life even early in the projects. But I think that in the phase 1 studies, we might not need to do that, but we might try to actually to deliver the product as fast as possible in a, of course, safe way to the patient and see if the project works.
Yeah, you're making a great point, and I think it's important to have a phase-appropriate approach, isn't it?
Yes, because again, the problem is that we never know if this therapeutic concept will work, and we spend so much time, so much efforts at the beginning of the project, and maybe then the project will be stopped because maybe there is no efficacy. So I think that we need to target the right efforts to put in the projects according to the phase in which we are. And this is true for any function, any steps of the process. So of course, people have a different way to see it, but I think as long as we deliver something that is safe for the patient, we are good.
I'm looking forward to further dive into today's topic, into developability and also formulatability. But before we do that, Giuseppe, let's talk about yourself, because your path from physical chemistry to computational structural biology is fascinating. So take us back to the beginning and tell us what sparked your interest, and then also what were some interesting pit stops along the way.
Yes, I think the beginning started in my undergrad studies when I started to have a look at this quantum mechanics, this theoretical chemistry. So I'm a chemist by education. And in those courses, I really discovered this fascinating capability of those techniques to predict properties of molecules without doing any experiment. So directly from computers, we could predict something out of the blue. And that was for me very fascinating and was the start of this kind of interest towards those in silico and computational methods. And I also had a very genuine interest in developing new drugs to help patients. And so I kind of tried to combine those two things together, and I got more and more interested in computer-aided drug discovery. Of course, I also worked in the lab during my undergrad period and also during my PhD, but then started more and more to lean towards the computational methods. And I think a very important pit stop in my life was a postdoc that I did during 3 years in the theoretical and computational biophysics group in Illinois at University of Urbana-Champaign. And there I really learned a lot of things and had a lot of experience in this field and really opened up a lot of different perspective for me. And probably that was one of the most important part of my career.
Can you paint us a picture, because you are combining, or let's say you are at the intersection of computational biology and now drug development or formulation development. What does a typical day look like? Are you sitting in front of the computer all the day and doing all kinds of modeling, or are you going to the lab? Is it a combination, or how does that look like?
Yeah, definitely what a computational chemist in a pharmaceutical company should not do is to stand just in front of a computer. Because I really believe that it's very important to exchange all the time with bench scientists. Because, of course, we need to understand what is the need, what is the most valuable thing that we could do for helping them in their day-to-day work. So my daily work is actually exchanging a lot with people in the lab and trying to understand what is the need and what I can support with the computational work. And then we decide, for example, we need to do a specific screening or we need to address a specific need of the project. And then I work maybe on my side to try to do this in silico assessment. And then I provide my final conclusion and recommendations. And then we exchange again. And then we can plan together also the activities in the lab. So it's really an exchange between the computational scientist and then the lab scientist.
Let's unpack this developability, formulation development, in silico approaches from the very beginning. Where do in silico approaches shine the brightest in drug development? I'd say from drug development, or even if I say drug development, that includes also, let's say, process development and the CMC part, because you've seen all kinds of different parts of the development of a biologic. So where do you see a particular benefit of these in silico approaches?
First of all, in silico approaches are really vast. So there is a lot of things that can be done, can be applied in pharma in general. I'm gonna kind of mention the approaches that are more related to what I do. So you define this concept of developability. So maybe not everyone is familiar with developability, but this is a new kind of a way of thinking. Because we want to develop drugs that are safe, efficacious, and manufacturable. And there is this concept of developability that was introduced a few years ago to try to select a candidate that has the highest possible developability. So from the experimental perspective, we can do a lot of experiments to understand how developable a drug is. However, we can also apply in silico methods to already screen properties of our API that can somehow predict the developability profile. So one regions of application for in silico methods is in the screening of different candidates. For example, in the final stage of discovery, where we have just a few candidates, like, I don't know, from 4 to 10. And in that case, the in silico methods can be very helpful in prioritizing candidates. So the one that might be more developable, more manufacturable. Once the final candidate is selected, so we start officially development. So in that case, we cannot change anymore the sequence. But in that case, we can also apply several in silico approaches to help develop, for example, the best formulation. In that case, you are not changing the sequence anymore, but you are changing whatever is around the API. So you might change, for example, the pH, or you might change the ionic strength or salts, surfactants, excipients. So in silico methods can be helpful in filtering out the conditions that might not be favorable for your API.
And that's such an important point, this developability, because for those who know me well, they know that I'm really thrilled to talk about this topic, because I'm a firm believer of starting CMC development early, already in discovery, and do this homework and look at these various properties to make sure that your molecule is developable and also, at the end of the day, manufacturable once you go into a larger scale. Can you tell us what is typically studied in the developability assessment? What kind of protein characteristics should you, as a minimum, look at, and then in order to make sure that your molecule is developable?
Yes, so there are several properties that we can predict. I can mention, for example, the hydrophobicity of the molecule. So we can predict if the molecule might have a specific peptide that is aggregation prone. And then we can mutate specific residues to remove these aggregation prone regions. Or we can predict the colloidal stability of the molecule. Typically, we can look, for example, at the charge of the molecule at different pH, and that gives us some idea of how stable might be the molecule in solution. We can typically also look at the chemical stability of the molecule, specifically looking at residues in the CDRs, so in the regions of the molecule that interact with the antigens. So those are very important regions for the efficacy of the antibody. We can also look into immunogenicity. There are several techniques that can predict these properties. So there are a bunch of things that can be predicted and can be very helpful in prioritizing the candidates. And you are really correct in saying that this must be done as early as possible, also having input from people from development. So I think it's really critical to have an exchange between research and development, because development people might also give some input about the formulatability of the molecule. So it's not only immunogenicity, but also if that specific candidate can be formulated in a specific conditions. So the exchange between research and development, I think, is really, really crucial.
Before we continue, biotech founders, I want to talk directly to you. Do you know that feeling when an investor asks about your manufacturing strategy and you freeze? One wrong CMC decision can cost you $2 million and 18 plus months of delays. Yet many CEOs are flying blind on these critical decisions. That's why I created the 1-week CMC strategy assessment. In 1 week, you get a personalized assessment of your molecule and development stage. We identify critical gaps before they become million-dollar mistakes, plus a strategic roadmap for high-stakes investor meetings. One CEO told me, "David took me from feeling powerless about our CMC roadmap to confidently pitching investors. An absolute game changer for time-pressed CEOs." Whether you are preparing for Series A, approaching IND filing, or facing scale-up challenges, this gives you confidence to ace investor and board conversations instead of surviving them. Ready to stop second-guessing your CMC strategy? Check the link in the show notes and secure your spot. Because your breakthrough therapy deserves a solid CMC strategy. Now, let's get back to our episode. Oh, yes, absolutely. You definitely speak my language here. It's absolutely crucial. And I've seen a lot of, unfortunately, projects where this has not been done or too late. And I've seen also consequences. So if scientists who is listening, start early and start, if you are in R&D, start communicating, talking to people then in process development and even people for manufacturing, that you have this input very early on. So I'm just curious about, let's take aggregation, for instance, and you do these in silico predictions. How accurate are these predictions, and how much do you still need to do in a wet lab to confirm your predictions?
Sure. So the predictions can be pretty accurate, but of course, there is no prediction that is always 100% accurate. It depends on the methods that you are using. And there are methods that are just sequence-based. So you don't need to have the structure of the antibody to predict the aggregation, for example. But then you can also look at the 3D structure of the protein, because as it's in a sequence, they will be in space. They can be close in space, and they might form some hydrophobic patches, for example, that you cannot really predict from the sequence. So this gives you additional informations. You can also consider different additional input to your predictions. So I think that one way to predict more accurately hydrophobicity is to take several informations from different methods and then combining all of them together in a holistic way to then conclude, okay, this molecule is more hydrophobic than this other molecule. So from my experience, I can tell that hydrophobicity is a parameter that can be predicted generally pretty accurately, but I would say that it's also difficult to estimate the hydrophobicity from the experimental perspective, because it's not always driven by hydrophobicity itself, but it's also driven by other factors like electrostatics. So it's also difficult to really quantify hydrophobicity from an experimental point of view. So that's something also to consider when we do comparison between what is calculated and what is seen in the experiments.
And I imagine that the more you do these experiments, the more data you generate from all kinds of different molecules, the better your predictions will become, because you can transfer knowledge, for instance, using hybrid models, or now probably also machine learning approaches, to then even better predict these results.
Exactly. If you use machine learning models, then you really need to have a significant amount of data, and then you can associate to those data several properties of the antibodies, also including the electrostatics contributions, and there maybe you can also increase your predictions. But maybe this is feasible. This is done already in several methods. I think that the most challenging part is to find actually the data itself, and also to find data that is representative of all possible API that we might have in development. Because nowadays we don't only have standard monoclonal antibodies, we have many multispecifics, we have ADCs, so we have many new modalities. So the issues with the machine learning is that once you have your training set on a certain categories, then your prediction might not really easily extrapolate on new modalities. So that's the reason why I also really like physics-based method, where you can also extrapolate. So you don't need really to have experimental data to train your model, but you just rely on the physics that is behind it, and you can still extrapolate on your new modality.
Your work has now evolved from developability, now focusing on the formulation development and also the formulatability. We'll get to the latter one in a bit. So I'm curious, how different is now your work, and I'd say your in silico approaches, as you're developing formulations? I mean, it's a very important part of the CMC development to have the right formulation. So yeah, let's start there. How different is this now from developability? And then we're going to move on to the second question, which it will be, what are these specific approaches to come up with a formulation that will work for your specific biologic?
Yeah, so the change in perspective is that we are going now from having several sequences in developability now to having only a single sequence. So that's the big change. In discovery, we have several sequences. Now we need to apply methods to select only one. Then in development, then we have only one selected. We cannot change this sequence anymore. So that's a very big change. And I think historically there has been a lot of work in the literature as well, like modifying, mutating the protein to try to improve the characteristic of the API. But then there is not so much in the literature. Once you fix the sequence, okay, how can we support formulation development now? So it feels that is not that much developed yet in the literature space. And this is now this kind of field that you are asking, this kind of formulation development, how can we support this effort with the in silico method? And now the idea is not to change the sequence, but to change whatever is around the protein. So the protein now is fixed, but the protein will feel a certain environment in a formulation. So it will experience a certain pH, a certain buffer species, certain salt or excipients, surfactants. So there are many things that might change the behavior of the protein. And I'm really convinced, and I have a lot of course, evidence that simulations and computational approaches can help in understanding what happens to the protein when we put it in a certain behavior. So that's somehow the change of perspective going from developability to formulation development in silico.
In the beginning of our conversation, you mentioned a phase-appropriate approach. So I'm curious now for the formulation development. How early should you start with that? And for instance, if we take a phase 1 development, should you just go with something that's quote off the shelf or everybody has done? Probably for an antibody, it's much easier. And then you put more effort into, or how do you see that?
I mean, like kind of platform approach can work if you have like a standard molecule, standard maps. But when you come to like a very complex multi-specific molecule, as we see like in the clinics now, that's more challenging, right? Your platform approach might not work, might or might not work. So I think that the in silico can still be very helpful in de-risking your strategy, in adapting your planning. So I would say that maybe you can have broad platform approach, and then maybe using computational methods, you can filter out conditions that might be less favorable for your specific molecule. So even for phase 1, you can still apply in silico methods to tweak a little bit your strategy. And the very nice advantage of computational methods is that you can apply them at any time. You don't need any material available. So they are relatively fast. So you can really start with this early for any phase. And of course, you can also do more for phase 2, phase 3, or any phase. If you want to do a much intense screening in the lab, of course, you can ask for more support from the computational guys. But again, any time you can always go to the in silico and ask for informations.
For those who are not familiar with formulation development, can you help us understand the difference between the formulation development and then you hear also the term formulatability? What is the difference and when should you do what, or is this something you do together?
I think formulatability is also a pretty recent term that was introduced somehow in parallel with developability to somehow test if a molecule can be easily formulated in development. So I think formulatability is something that comes together somehow with the developability when you screen your candidates before starting development. And it gives you this kind of perspective from development because you are looking forward and into the formulation development, and then you are asking, is this molecule feasible for a certain formulation, or might this molecule be challenging to formulate? So formulatability is kind of trying to address this question, and formulation development instead is a work package that instead is executed during development in the drug product development group. And it's, of course, the task where we try to then find the best suitable formulation for a certain API. Having all prior knowledge or any type of information can be helpful to really set up all the experiments.
That wraps up part 1 of our conversation with Giuseppe Licari. We've explored how computational methods are revolutionizing developability assessments and catching formulation problems early. In part 2, we'll dive deeper into excipient selection and real-world integration strategies. If you're finding value in these insights, please leave a review on Apple Podcasts or your favorite platform. It helps other scientists like you discover these conversations. See you next time in part 2. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.